everybody, and welcome to Science with the Bish Talks Eukaryotic Cells. The cell, nature's brilliant idea to pack all the important bits into a microscopic bubble, and just hope nothing leaks. In this video, we'll be looking at the main organelles found inside eukaryotes and what they do, how their structure helps them to do their jobs, and a few key differences between plants and animal cells. So let's begin. All cells are surrounded by a cell surface membrane. It keeps the inside of the cell separate from the outside world, like a very slimy front door. It controls what enters and leaves the cell, allowing some substances through and not others. We call that
partially permeable.
The membrane itself is made of a phospholipid bilayer, a double layer about 10 nanometers thick. The hydrophilic phosphate heads are sticking out of the membrane, and the hydrophobic tails point into the membrane. Here's a quick summary for all those taking notes. The nucleus is one of the biggest features inside any eukaryotic cell. It's separated from the rest of the cytoplasm by a double membrane, known as
the nuclear envelope,
which is filled with pores that work like tiny doors for molecules to pass in and out. Inside, the nucleus holds the cell's DNA, all packed up into structures called chromosomes. These chromosomes are made from DNA wrapped around proteins called histones, and together they're known as
chromatin.
Basically, genetic spaghetti. And in the middle, you'll find the nucleolus, a busy little region where ribosomes are produced before they're sent out to do the real work.
Protein synthesis.
Here's a brief summary of the nucleus. Next, the endoplasmic reticulum. It comes in two flavors, rough and smooth. The rough endoplasmic reticulum, or RER, is covered in ATS ribosomes and found attached to the nuclear envelope, giving it a texture that looks, you guessed it, rough. Its job is to fold and package proteins that have just been synthesized by those ribosomes. Once ready, the proteins are sent off to other parts of the cell, usually the Golgi body, for further tweaking. The smooth endoplasmic reticulum, or SER, doesn't have any ribosomes attached. It handles a different set of tasks, mainly making lipids and steroid hormones. So, rough ER builds the proteins, smooth ER builds lipids. Here's a summary for the smooth and rough endoplasmic reticulum. Let's talk about the Golgi body, sometimes called the Golgi Apparatus or Golgi complex, depending on how fancy you're feeling. It's a stack of flattened membrane-bound sacs, a bit like a pile of pancakes, if pancakes could process proteins. Proteins made in the rough endoplasmic reticulum arrive here next. The Golgi modifies them, adding sugar groups or changing their shape, then packages them into small vesicles for transport to wherever they're needed, inside or outside the cell. In short, the Golgi is a cell's post office. It sorts, packs, and delivers. And hopefully doesn't lose your parcels. Here's a summary for the Golgi apparatus. Next up, mitochondria. You've probably heard them described as
the powerhouse of the cell.
And for once, that's not just textbook dramatics. Mitochondria are oval-shaped organelles with a double membrane. The inner membrane is folded into shelves called cristae, which increase the surface area for the chemical reactions that release energy from glucose. And this is used to make ATP, the cell's main energy carrier. Inside the inner membrane is a fluid called the matrix, which contains enzymes for respiration, a small amount of circular DNA, and 70S ribosomes, the smaller type also found in prokaryotic cells. So, in short, they're the cell's energy centers, turning food into usable ATP, so everything else can function. Here's a summary for the mitochondria. Now, let's talk about ribosomes. Small, round structures that do one very important job. They build proteins, a process known as
protein synthesis.
You'll find them floating freely in the cytoplasm or attached to the rough endoplasmic reticulum. They read messenger RNA and join amino acids together in the right order. In eukaryotic cells, most ribosomes are ATS, the larger type found in the cytoplasm. But inside mitochondria and chloroplasts, you'll find 70S ribosomes, smaller ones that look suspiciously like those in prokaryotes. A little reminder of their ancestral origins. Here's a summary for the ribosomes. Now, let's talk about vesicles and lysosomes. Two small but essential organelles that keep the cell organized and functioning. Vesicles are small membrane-bound sacs used for transport and storage. They're formed when sections of the Golgi body pinch off at the ends, producing packages that carry molecules around the cell. They can fuse with the cell surface membrane to release their contents outside the cell. That's
Exocytosis.
Or bud inwards from the membrane to bring material in. That's
Endocytosis.
Some vesicles contain hydrolytic enzymes, which are used to break down biological molecules, waste materials, and damaged organelles. These enzyme-filled vesicles are known as
Lysosomes.
lysosomes, also help destroy pathogens engulfed during phagocytosis, and break down cell components during apoptosis, controlled cell death, or the cell's way of bowing out gracefully. In short, vesicles move material around the cell, and lysosomes handle the cleaning and recycling. Together, they keep everything running smoothly. Here's a summary for vesicles and lysosomes. Next up, centrioles and microtubules. These two work closely together to keep the cell structured and organized, kind of like the cell's internal scaffolding and construction crew. Centrioles are short, hollow cylinders made from microtubules. They come in pairs. Two centrioles at right angles form a centrosome. These play a key role in cell division by organizing the spindle fibers that separate chromosomes. You'll find them in most animal cells, but not in flowering plants or fungi. They seem to manage just fine without them. Microtubules, on the other hand, are hollow tubes built from tubulin protein. Each microtubule is made when alpha and beta tubulin molecules join to form dimers, which then link together into long chains called protofilaments. 13 of these form a cylinder, and there you have a microtubule. They make up the cell cytoskeleton, provide both shape and support, and help with the movement of organelles. Here's a summary of centrioles and microtubules. Next, Cilia and Microvilli, two tiny projections that live on the cell's surface, each with a very different job. Cilia are hair-like extensions made from microtubules arranged in a 9+2 pattern. That's 9 pairs around the edge and 2 single microtubules in the middle. This arrangement allows them to move rhythmically, pushing substances along the surface of the cell. Microvilli, on the other hand, are much shorter and don't move at all. They're simple folds of the cell surface membrane that increase the surface area to make absorption more efficient. You'll find them in areas that specialize in absorbing substances, such as the small intestine. Here's a summary of cilia and microvilli. Now, let's look at chloroplasts, the structures that let plants turn sunlight into something useful. They're larger than mitochondria, and like them, are enclosed by a double membrane that holds everything together. Inside are flattened membrane sacs called
Thylakoids.
stacked into towers known as grana. These stacks are linked by lamellae, thin membranes that connect the grana and help move materials between them. The thylakoid membranes contain chlorophyll and other photosynthetic pigments that absorb light energy needed for photosynthesis. The fluid in the chloroplast is known as stroma. Floating in the stroma, you can also find circular DNA molecules and 70S ribosomes. Here's a summary of the chloroplast. Next up, the cell wall. It sits outside the cell surface membrane and gives the cell its shape and strength. In plants, the wall is built from the polysaccharide cellulose, long chains of beta-glucose molecules that form a tough fibrous network. In fungi, the same job is done by chitin instead of cellulose, but the idea is the same: structural support and protection. Unlike the cell membrane, the cell wall is freely permeable, meaning water and dissolved substances can pass through without any special permission. Here's a quick summary for all those taking notes. Now, let's talk about Plasmodesmata. These are tiny channels that connect one plant cell to another, like little bridges made of cytoplasm. They pass straight through the cell wall, allowing water, ions, and small molecules to move directly between neighboring cells. This means plant cells can share resources and communicate, making the whole tissue function as one big, interconnected network. Here's a quick summary for all those taking notes. Finally, the large permanent vacuole and the tonoplast. In plant cells, the vacuole is a large fluid-filled space that takes up a good portion of the cell's interior. It's filled with cell sap, a mixture of water, ions, sugars, and other substances. And it helps to keep the cell firm by maintaining
Toner pressure.
The vacuole is enclosed by a membrane called the tonoplast, which is partially permeable and controls what enters and leaves the vacuole. Here's a quick summary for all those taking notes. Thank you very much for watching. If you made it this far, please take a moment to like and subscribe. But most of all, love to your mothers.